This is part 2 of the Chapter 10 recordings on photosynthesis. And I'm going to skip some of this. Let's see. Because you don't need to know all of it. But this does show you what we call the absorption spectrum. So every pigment has a different absorption spectrum. And where the peak is, that's where it absorbs. That's the wavelengths that it absorbs best. So we look at chlorophyll A. From what I have seen, chlorophyll A is a dark, it's like not a dark green, but it's like a regular green, like just green, Kelly green, you know. In real life, to me, the pigment chlorophyll A is just plain green. And so chlorophyll A absorbs best right here. I guess you would say like around 440. And then it also absorbs best at around 680 nanometers of light. So it absorbs best in the indigo region, like bluish purple region, and in the red-orange region. And then chlorophyll B is similar, but it's the wavelengths it absorbs are a little bit different. So chlorophyll B is an olive green, and it absorbs more on the blue end, closer to 500, and it absorbs more on the orange end, closer to 600. Then you have the carotenoids. The carotenoids really pretty much don't absorb from this point on. So carotenoids are going to absorb closer to the green end, maybe the blue and the green end of the spectrum. And the reason for having different pigments is for that reason. It's so that you can have a greater, more wavelengths of light absorbed from the sun, and so more efficient photosynthesis. Here's a good way to say it. The accessory pigments, such as chlorophyll B and the carotenoids, broaden the spectrum used for photosynthesis. So they allow for a greater amount of visible light to be absorbed. All right. What I'd like to see is, like I did with the electron transport chain, I'd like to talk to you about the illustration first, and then we'll go back to the notes. So here's what we're looking at. We're looking at this phospholipid bilayer is the membrane of a thylakoid. It's the thylakoid membrane. And inside here is the space inside the thylakoid. And outside is the stroma of the chloroplast. So we're inside of a chloroplast. This is the thylakoid membrane. And this is what happens in the light reactions of photosynthesis. So you have this photosystem. It just looks like a really, really large protein, membrane protein. And it is. It's a complex of molecules and a very, very large protein. So within a photosystem, you're going to have light harvesting complexes that consist of pigment molecules. And those pigment molecules are going to absorb photons of light. Notice that's photon, not proton. Photons of light from the sun. And that's going to excite an electron in whatever, all of these pigment molecules in this complex, but the electron is finally going to end up being captured by a special pair of chlorophyll A molecules. So they're called the reaction center chlorophyll A molecules. Then the electron is going to be next captured by the primary electron acceptor molecule. And at that point, it's going to be used to generate NADPH and ATP in a process like that involves an electron transport chain. So, a photosystem is a reaction center complex, and then a bunch of pigment molecules, which we call light harvesting complexes. And the reaction center complex is basically going to have the chlorophyll A molecules that are called the reaction center chlorophyll A molecules, and then a molecule called the primary electron acceptor. And then the light harvesting complexes are going to contain pigments, including chlorophyll A, but it can be chlorophyll B and carotenoids also. The primary electron acceptor, remember, is included in the reaction center, and it's basically, it accepts excited electrons that come from the chlorophyll A molecules. So again, sunlight is absorbed by the pigment molecules in the light harvesting complexes, and then an electron is excited and it kind of bounces around from pigment to pigment, and it ends up in these special chlorophyll A molecules. The chlorophyll A molecules are able to excite the electron, in other words, raise the energy of the electron, and the electron is next accepted by the primary electron acceptor. There are two photosystems. There's photosystem 2, and we're doing them backwards because photosystem 2 comes first in the thylakoid membrane, and then photosystem 1. So photosystem 2 is called P680 because its reaction center chlorophyll A is best at absorbing light at 680 nanometers. And photosystem 1 is called P700 because its reaction center chlorophyll is best at absorbing 700 nanometers. There are 8 steps in linear electron flow through during the light reactions, basically. All right, the first step is a photon of light is absorbed by a pigment in the light harvesting complex. So photon hits a pigment, and its energy is passed among pigment molecules. So basically, it causes an electron to bounce from one pigment to another among the light harvesting complex. Then, in step 2, because the last place that electron will arrive is at a pigment called chlorophyll A, special chlorophyll A molecules, which are going to excite the electron. So the excited electron from photosystem 2, or P680, is transferred to the primary electron acceptor. And then an enzyme splits water. So the water molecule that comes in, through the roots of the plant, that water molecule is split, and it ends up providing 2 electrons, 2 hydrogen ions, or protons, and an oxygen atom. The electrons are used in the electron transport chain. The hydrogen ions are used during chemiosmosis. So electrons and hydrogen ions are then used to make the ATP and NADPH. Now, the oxygen atom ends up combining with another oxygen atom and forms oxygen gas, which is O2. All right, for electrons, the primary electron acceptor of photosystem 2 releases an electron, and it is passed from electron carrier to electron carrier. And as it is passed from carrier to carrier, it releases energy. So the energy released by electron transfer is used to pump hydrogen ions into the thylakoid space. This should sound familiar. This is the electron transport chain and chemiosmosis. We have already learned about this in Chapter 9, it is not any different. So the energy from the flow of electrons is used to pump hydrogen ions across the thylakoid membrane into the thylakoid space. And that creates a concentration gradient of hydrogen ions inside the thylakoid. And we have that energy is used to produce ATP. And we call that chemiosmosis. And then the electron is transferred to photosystem 1, or P700. And that electron receives energy from sunlight, and it is taken up by the primary electron acceptor, and then it is transported down and passed down an electron transport chain again. The energy from the electron is used, energy from the electrons is used to generate NADPH. So there's not an electron transport chain after photosystem 1, but the electrons are still used to form NADPH. So our final electron acceptor in the case of photosynthesis is NADPH. Remember, in cellular respiration, our final electron acceptor was oxygen. So our final electron acceptor is NADPH. Now that ATP has been produced and NADPH has been produced during the light reactions, they're going to enter the Calvin cycle. All right, going back to the linear flow, or what we call the non-cyclic cycle, you have photosystem 2. Sunlight is absorbed by the pigment complex, and it's used to excite electrons and an electron, and the electron is going to bounce from pigment to pigment, and it's going to be taken up by chlorophyll A molecules, which are going to excite the electron, and it will then be taken up by the primary electron acceptor, sent down an electron transport chain, and then the energy from the electron transport chain plus chemiosmosis of the hydrogen ions is used to make ATP. So ATP is produced by photosystem 2. Then that electron ends up in the chlorophyll A molecules of photosystem 1. Sunlight excites pigment molecules in photosystem 1, and whatever electron that you see bouncing around is going to replace the electron that leaves from the chlorophyll A molecules. But the electron is going to end up in the primary electron acceptor, and then it's going to be used to produce NADPH. There is no electronic transport chain after the, it says that, but it's not truly an electron transport chain and chemiosmosis. It's just going to be electrons are, they just are transported to this one molecule, whatever FD means, instead of a chain, But anyway, the electrons are going to combine with hydrogen ions and NADP plus to produce NADPH. So NADPH is produced by photosystem 1. Now, another thing that's important to understand is that where do the electrons come from to replace the electron that's lost from photosystem 2? And the answer is water. The splitting of water replaces the electrons that are lost from photosystem 2. Now, there is another mechanism, I guess you would say, for the light reactions. And instead of being linear or non-cyclic, this is called cyclic electron flow. And basically, this only happens with photosystem 1. So when this process occurs, rather than the non-cyclic or linear electron flow that used both photosystems, this process only produces ATP. So ATP can be produced by photosystem 1, but only in cyclic electron flow when the photosystem 2 is not involved. When photosystem 2 is involved, then photosystem 2 produces ATP, and photosystem 1 actually produces NADPH. So the overview for both types of, or both versions of light reactions, the version that uses both photosystems, photosystem 1 produces ATP, and photosystem 2 produces ATP, and photosystem 1 produces NADPH. When photosystem 2 is not involved and it's only photosystem 1, we call it cyclic electron flow, and photosystem 1 actually generates ATP in that case instead of NADPH. Oh, sorry. Okay, comparing chloroplast and mitochondria, because the chloroplast carries out photosynthesis and it does involve an electron transport chain. Mitochondria carries out cellular respiration and it also involves an electron transport chain. So we're going to compare them. Both these organelles generate ATP by chemiosmosis, which is, the use of the electron transport chain to pump hydrogen ions across a membrane and produce ATP that way through oxidative phosphorylation. The chloroplast is not called oxidative phosphorylation, it's called photophosphorylation, but it's the same thing. And it tells you here how photophosphorylation differs from oxidative phosphorylation. For one thing, the electrons are replaced by water molecules in photophosphorylation. The electrons that are excited and used in the electron transport chain, they are replaced by the splitting of water, while in the mitochondria and cellular respiration, the electrons are replaced by, it says they're extracted from organic molecules, but basically they come from the NADH and the FADH2. And mitochondria transfer chemical energy from food to ATP, whereas chloroplasts transform light energy to ATP. So in the mitochondria, its chemical energy is transferred to produce ATP, and in chloroplast, it is light energy that is used to generate ATP. That's just a little comparison. And the other comparison just has to do which membrane is used during electron transport and which direction the hydrogen ions are pumped, but I don't think that matters that much as long as you understand they're pumped across the membrane. The hydrogen ion concentration builds up on one side of the membrane, and then as it flows back through ATP synthase is used to generate ATP. So it doesn't really matter which side of the membrane that hydrogen builds up. It's going to flow to the other side through ATP synthase and generate ATP. Now, ATP and NADPH are produced in the light reactions, and they're going to be the fuel for the Calvin cycle reactions. And this is a picture of a larger portion of the thylakoid membrane, so you can see the light reactions a little bit better. In this picture, you have photosystem 2 and photosystem 1. So basically, you're going to have photosystem 2 generating ATP right there. And you have photosystem 1 generating NADPH. And both enter the Calvin cycle. So there's an animation on the light reactions. I would encourage you to watch this one. It's slide number 75. And another one, slide 76. And then now we've come to the Calvin cycle. So this is slide 77, and this is where I will start the next recording.